Hello friends, I am Dr. Kalpana and I am here for NEET Biology. The topic I am taking today is structure of DNA. DNA or deoxyribonucleic acid is genetic material in most of the organisms. The main components of DNA are nitrogenous bases, deoxyribose sugar and phosphoric acid. When a nitrogenous base combines with deoxyribose sugar, we get nucleoside. And when a phosphate group joins nucleoside, then we get nucleotide. Many such nucleotides join and then we get polynucleotide chain. Two such polynucleotide chain, this is one nucleotide chain, polynucleotide chain, this is another polynucleotide chain. Then they coil and we get the double helical structure of DNA. So this is double helix of DNA. Now we will take these components one by one in detail. First the nitrogenous bases. So first component of DNA is nitrogenous bases. Nitrogenous bases are the nitrogen containing compounds and are of two types in DNA. First are called purines, in which we have adenine and guanine. Second types are called pyrimidines, in which we have cytosine and thymine. Now the purines. First type of nitrogenous bases are purines. Purines are the compounds which have two rings. One 6-membered pyrimidine ring and another 5-membered imidazole ring fused together. This is imidazole ring. In purines, 4 nitrogen atoms are present at 1, 3, 7 and 9 positions. Numbering of atoms, we start from here 1, 2, 3, 4, 5, 6 and then in the second ring we proceed in anti-clockwise manner 7, 8 and 9. So 6-membered ring and 5-membered ring fused together and 4 nitrogen atoms present at 1, 3, 7 and 9 positions. And if it is adenine, in adenine one NH2 group is here and H attached with this. So for balancing we have to put the double bonds. This is adenine. In short, it is written as A. The molecular formula is C5H5N5. Now the second purine is guanine. Again here we have a 6-membered ring, pyrimidine ring, fused with a 5-membered ring, imidazole ring, with nitrogen present at 4 places 1, 3, 7 and 9 positions. 1, 2, 3, 4, 5, 6, 7, 8 and 9 positions. In guanine we have double bond O here, NH here and one NH2 attached here. To balance we have to put the double bonds like this. So this is guanine. Represented as G. Molecular formula is C5H5N5O. So these are the two purines. Then the pyrimidines. In pyrimides we have only one ring, 6-membered ring with nitrogen present at only 2 places. One and 3. Here we start from here, numbering start from here, 1, 2, 3, 4, 5, 6. So nitrogen present at 1 and 3 places. If it is cytosine, double bond O is here, and to balance we have to put the double bonds like this. This is cytosine, represented in short as C, and the molecular formula is C4H5N3O. Then the second pyrimidine is thymine. Here also we have only one 6-membered ring with nitrogen present at 1 and 3 places. It is 1, 2, 3, 4, 5 and 6. In thymine it is double bond O here, double bond O here, H here, H here and one methyl group attached here. And to balance we have to put only one double bond. This is thymine, represented as T, and the molecular formula is C5H6N2O2. So these are the 4 nitrogenous bases. Then the second component of DNA is sugar, and that sugar here is deoxyribose sugar. Deoxy means one oxygen less than ribose. Ribose is a 5-carbon compound, and if one oxygen is less in ribose, then it becomes the deoxyribose sugar. So second is deoxyribose sugar. This is 5-carbon compound. One, 2, 3, 4 and 5. So 5 carbon are there, and everywhere it is HOH. Here also HOH, but at position number 2 instead of OH we have only H. So here only HH, otherwise it is HOH, HOH, like this. So this is deoxy, and oxygen is less at position number 2. This is deoxyribose sugar. The third component is phosphoric acid, which gives the phosphate group to the DNA and is responsible for the acidic nature of DNA. So phosphoric acid The formula is this. So these are the three components. Now we will see the linkages between these. How the nitrogenous base links with sugar, or how the sugar links with phosphate, like that. So first, the linkage between a nitrogenous base and deoxyribose sugar. So the linkage between a nitrogenous base and deoxyribose sugar. The linkage between a nitrogenous base and deoxyribose sugar is called the beta N-glycosidic linkage. For example, we take the example of adenine, a nitrogenous base, and how it combines with deoxyribose. So this is adenine, and numbering is 1, 2, 3, 4, 5, 6, 7, 8 and 9. In case of purine, sugar gets attached with the nitrogen at position number 9. That is the difference. In case of pyrimidine, it is the first nitrogen at position of 1, but in case of purines, it is nitrogen at position number 1, position number 9. And here we will make the sugar. This is 1, 2, 3, 4 and 5. This is deoxyribose sugar. So what happens when a nitrogenous base comes in contact near the deoxyribose sugar? This H attached with the N9 and this OH attached with the C1. They form water, and then water is liberated. So they get directly linked and minus H2O. H from here and OH from here. So water is formed and nitrogenous base, this is adenine and this is deoxyribose sugar. So in this way it is connected, N9 and C1, and this bond is called the beta N-glycosidic linkage. Thus a nucleoside is formed. If it is adenine, then the nucleoside form is called deoxyadenosine. And if it is guanine, then it will be deoxyguanosine. The linkage is beta and glycosidic bond, because if a bond is formed between a molecule and a sugar, then it is glycosidic. Here it is sugar, so it is glycosidic. It combines with the nitrogen of this compound, so it is N-glycosidic. And beta, because the OH attached with the C1 of sugar, is above the plane of the sugar. Suppose this is the plane of the sugar, then OH lies here above it. So it is called beta N-glycosidic linkage or bond. In case of pyrimidine, for example, we take the cytosine example of cytosine. Nitrogen present at two places. This is cytosine and numbering is like this: 1, 2, 3, 4, 5 and 6. Here is sugar. This is deoxyribose sugar. Again what happens? This H and this OH. H attached with the N1 and OH attached with the C1. This is 1 carbon, 2, 3, 4 and 5. So N1 and C1, they combine and a molecule of water is liberated. And N gets directly combined with C. This bond is beta N-glycosidic linkage. So that is the difference between purine and pyrimidine. Here it is N9 and here it is N1. In this case, the nucleoside form is called deoxycytidine. Similarly with thymine, it will be deoxythymidine. So deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. These are the 4 nucleoside forms when a nitrogenous base combines with sugar and we get the nucleoside, and the linkage is beta N-glycosidic linkage. Now what happens? Phosphate. Now the phosphate combines with the nucleoside. Phosphate group combines with the nucleoside and we get the nucleotide. So now the linkage of phosphate group with nucleoside. Actually it is the sugar molecule of nucleoside. Phosphate combines with the sugar molecule of nucleoside. This is sugar, this is phosphate, phosphoric acid, and here is the sugar, CH2OH, OH, OH. This is 1, 2, 3, 4 and 5. This is sugar, deoxyribose sugar. So what happens in this case? This H and this OH attached with the carbon number 5 of sugar. They form water. Water is liberated and the phosphate group gets directly linked with carbon at position number 5 of sugar. And as I told you, here at position number 1, it remains attached with the nitrogenous base. At position number 9, if it is purine, and this bond that is formed here is ester bond. It is called the phosphoester bond. you know ester is formed when alcohol combined with sugar, acid, OH of an acid. So here is the OH and here is the H. So it is phosphoester bond, and here is the beta N-glycosidic linkage. It is the purine. Similarly, another nucleotide can also be formed with pyrimidine also. It is like this. P double bond O, OH, OH, OH. Then we have the sugar here, C, H2, OH. Here OH, OH. Numbering is 1, 2, 3, 4 and 5. And in this case, here it will combine with the pyrimidine. It may be cytosine or thymine, and it will be position number 1 by beta and glycosidic linkage. Then what happens? This H and this OH form the water, and this phosphate group get attached with the carbon. So it get attached with the carbon number 5 by phosphoester bond. This is one nucleotide where we have purines, and here it is pyrimidine, the nucleotide form. This is also the phosphoester bond. Now in this way nucleotides are formed. Now I told you that nucleotide they join, and then we get the polynucleotide chain. So how is this formed? Formation of polynucleotide. Any two nucleotides can join forming dinucleotides, and if the nucleotides go on joining one another, then it will become polynucleotide. How the two nucleotides join? Here you will see this is OH attached with this polynucleotide at position number 3, and here is the OH attached with phosphate group that is attached with carbon number 5 of sugar. Then this OH and this H combine, and then they form water. Water is liberated, and this sugar get attached with the phosphate of the next nucleotide. This is OH of sugar at position number 3. This is phosphate attached with carbon at position number 5 of the next nucleotide. This is also phosphoester bond. 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 But what is the difference? Here the carbon number 3 is involved in this, so it is called the 3-phosphoester bond. And here carbon number 5 of sugar is involved, so it is called 5-phosphoester bond. And this bond total will be called 3,5-phosphodiester bond. Thus a dinucleotide is formed by 3,5-phosphodiester bond. It means on one side the phosphate group will combine with the position number 3 of sugar, and on the other side it combines with the carbon number 5 of sugar. So this is 3,5-phosphodiester bond. Like this it will go on. If the next polynucleotide is there, then it will combine with the OH at position number 3, and then the phosphate will combine with position number 5. Again this way it will form polynucleotide chain. But what will happen? So we can represent it as this. phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. In this way, polynucleotide chain will be formed and sugar get attached with, suppose it is adenine, it is thymine, any nucleotide can be there, nitrogen base can be there. So in this way, a polynucleotide chain will be formed. These bonds will be beta N-glycosidic and these bonds will be phosphoester bonds. But in a polynucleotide chain, at one end there will be free phosphate group attached with the carbon 5 of the sugar. So this end is called the 3 end of polynucleotide. 5 end. Why 5 end? Because this phosphate is attached with the 5 carbon of this sugar. And at the other end, what will happen? We will have OH at position number 3 of sugar free. Free OH at position number 3 of sugar. So this end is called the 3 end. This is very important. 5 end means free phosphate attached with position number 5 of sugar. 3 end means free OH at position number 3 of sugar. So in this way, each polynucleotide will have one 5 end and one 3 end. Now, double helix. I told you 2 polynucleotides chain, they coil and we get the double helical structure of DNA. Formation of double helix. The double helical structure of DNA was given by 2 scientists in 1953. James Watson and Francis Crick proposed the double helical nature of DNA, and their model was based on the X-ray diffraction data of Morris Wilkins and Rosalind Franklin. So the double helical structure is given by the Watson and Crick. This is Watson and Crick model of DNA, and their model was based on the data of X-ray diffraction data of Morris Wilkins and Rosalind Franklin. And the main points of, according to them, the DNA double helix consists of 2 polynucleotide chains where the nitrogen bases are attached to one another with hydrogen bonds, and the 2 polynucleotide chains are complementary. Complementary means if we know the sequence of bases, nitrogenous bases in one chain, then we can predict the sequence of nitrogen bases in the second chain. How this we will see. The main first point of this double helical structure is that the backbone of the double helix is made up of phosphate sugar, and the nitrogenous bases project like this. This is one polypeptide chain and this is another. So these are the 2 chains, and these chains are anti-parallel. The nitrogen base is the backbone made up of phosphate sugar, and the nitrogen bases they project inward. If the polarity in one chain is 5-3, it is 5-3 because we have phosphate here and free OH here, so it is 5-3. Then in the second complementary this strain ring, we will have the 3-5 polarity. So in this polarity is 5-3 and in this it is 3-5. If here we have free phosphate, then we will have free phosphate here. We have OH, free OH here, then we will have free OH here. Then the nitrogenous bases of these strings, they get attached by hydrogen bonds. Adenine always forms hydrogen bond, 2 hydrogen bonds with thymine and vice versa. Means if A here, T here, then T here, A here. Similarly, guanine forms 3 hydrogen bonds with cytosine of other string and vice versa. So C and G will form 3 hydrogen bonds and they will form similar. And these two, they are called base pairs. Everywhere you can see there is one purine, one pyrimidine, purine, pyrimidine. Means one double ring structure, one single, one double. This way the distance between the two chains remains the same throughout its length. Then how this form, the hydrogen bonds are formed, that we will see. Adenine and thymine form 2 hydrogen bonds, and thymine, guanine and cytosine, they form 3 hydrogen bonds. Hydrogen bond is formed when hydrogen gets attached with on one side with covalently with one electron